Good afternoon. This is the first lecture in our online teaching process. We will speak about the feeding of laboratory animals. This is in the series of standardization. Standardization of the animal which is treated as a black box. The black box hypothesis shows us that we treat the animal as a black box and therefore all the other circumstances should be standardized. This is what we have considered that the genetic standardization, that the sex standardization, that the level of uh, pain and suffering, the level of stress, the environment, etc. But after that, we can tell that the experimental stimuli are the only differences between control and experimental group. Therefore, the biological answer between the difference of the biological answer between the experimental group and the control group owed to our experimental stimuli. Therefore, we had to make all these standardization included the feeding of the laboratory animals. Uh, Next slide, which is the take-home message at the beginning. Yes, because it is at the same time a little bit a study plan. The nutrient requirement are for longevity and reproduction instead of maximum production. The diets may be not only natural but also semi-synthetic and synthetic. Using sterilization and heat treatment, part of the vitamins will be destroyed. The physical forms may be, besides the pellets, gel, liquid form too, and consideration of breed, strain, especially if exotic or transgenic animal is used, is important, as well as the consideration of the lack or presence of microbiota or microbiome. The feeding is mostly ad libitum, but we have new tendencies. And consideration of stress, microbiome, and the circadian rhythm is also important, but it's depend upon the time that how detailed can be treated. Here you see a nude mouse. This is the NUNU homozygous recessive mutant and its litter. There is no SCID or serious combined immunodeficiency mouse on the picture, but we know that there is. You see a marmoset monkey. You can see that even in this picture, the consideration of species and strain is really essential. You see also great differences on this next picture from the iguana up to the gerbil through the Japanese quail and hamster. A series of different species are present as experimental animals. To make some classification anyway, we can classify these animals like herbivore, included fruit eaters like the rabbits, guinea pigs, dagus, chinchillas, marmots, flying dogs, prairie dogs, and small ruminants. Omnivore may be the burunduk or striped chipmunk from Siberia, the mice, hamster, gerbil, rats, voles, and mini pig and pig. And carnivores included the insectivore may be the tenrec, the hedgehog, the ferrets, the dog, and the cat. On this real picture, you can see the more digestive system. You can see a nice uh, uh, double stomach, very simple small intestine, a virtual cecum in the middle, and a simple colon. This quite double stomach is very similar to the today's guinea pig's stomach. The picture shows us different form of insectivores because this ancestor of the mammals, which was similar to that, was also insectivore. Here you see the today's insectivores, the human, the ant eaters, and other animals. The digestive system of the different experimental animals shows us a lot of characteristics of the digestion. Digestive system of the dog, and here you see on the picture the most important breed which is used in the experimental purposes, the beagle. We have a small, at about 8 kilogram live weight, and a larger form of 12 kilogram of body weight. In the digestive system picture, you can see a huge stomach and a small and simple intestine with a very, very small virtual cecum. Our conclusion can be that it is sufficient to feed the dog once a day. And the second, that there is no real fiber digestion. The digestive system of the rat picture shows us a real and artistic picture. You can see on the real picture that there is a liver, and below the liver there is a stomach. After you have the duodenum, jejunum, and ileum. After you have a medium to big cecum and colon. This is an absolute omnivore type of digestive system. On the next slide, you can see as a mini pig on a dog competition, but this is only as a companion animal. But these pot-bellied mini pigs. But in the laboratory, we use Göttingen mini pig, Yucatan mini pig. And the digestive system is also an omnivore one, with a medium-sized stomach, a medium-sized cecum, medium-sized colon, of large colon, and a long small intestine. What is not in the picture that the pancreatic enzyme activity is really high, so they can digest the starch very effectively, little bit more effectively than the human. And the digestive system of the ruminant shows us the rumen and the very long small intestine and medium-sized cecum and colon. And finally, you see a picture different types of rodents, because from now on we will concentrate on the feeding of the rodents, because all the other animals will be treated in details during the animal nutrition course. On the picture on the left up, you see a obese and and the normal inbred mouse. You see a wild Norwegian rat, the left corner, and a Swiss mouse and a Swiss cheese joke, only to remember for you that the Swiss strain is an important strain for the laboratory mouse. The psychotrophy or coprophagy is a very important feature of most of these animals, included the rabbit, guinea pig, chinchilla, dagu, gerbil, and hamster. Yes, the phenomenon of trophallaxis. The phenomenon of trophallaxis is the use of own or species companion biological product like feces, secrete, saliva for feeding. It means that the ants, when they use small lice and by tickling they receive some milk-like secrete. This is like our cow keeping. This is not a trophallaxis. Own or species companion biological product. The example is the coprophagy. Coprophagy is the eating of the real normal feces, which is typical in case of real energy and protein deficiency. For example, rats and even horses are eating their own feces if they receive a feed mixture which contains less than 10% protein. This is the maintenance level. Psychotrophy is a very special form of the coprophagy when two types of feces is excreted and the cecum content is excreted in a different time than the hard feces. For example, on the picture on your right, you can see a rabbit who is just taking off the soft feces from the anus directly. This is very efficient way of the trophallaxis because the psychotrophy, this soft feces, contains a lot of protein and vitamins of the group B and K. The crop milk of the pigeon and the crop milk of the female parrots is also a type of trophallaxis, similarly to the vomit or puke of the canidae, which was in the wilderness that the hunting individuals carried home the meat in the stomach and vomited to the lactating bitches this one. Today we can see in some kennels the so-called the jealousy vomiting, when animals eat up more and more from the pet food and after vomiting in a silent corner to eat it alone. So this is this slide, and the next slide shows us the rabbit 2 feces type. The cluster-like is the psychotroph or soft feces, and the individual yellow balls are the hard feces, which is low in protein and high in fiber. The next picture shows us the guinea pigs, two types of feces. On the left side you see the yellow croissant-like normal feces. On the right side you see the black, smaller cucumber seed-like psychotroph content. The only difference with the rabbit is that the guinea pig is eating it from the button, from the cage and not from the anus. And only to show the two types of chinchilla faces. Left is the cecum content, which is black and high in protein, and right you will see the yellow and normal feces, low in protein and high in fiber. How to determine the nutritional needs of the laboratory animals? The criteria of requirement evaluation. Could it be a growth? Do we eat laboratory animals? No. Production? Is there a milk production or so? No. These two criteria is for the production animals, for slaughtering pig, a broiler chicken, etc., milking cow. But in case of the laboratory animals, we use the reproductive capacity, and the longevity. How long is the lifespan? As long as possible, to make possible to extrapolate data for humans, human aging research, for example. The formulation of the laboratory animal feed can be in way of open formula or closed formula. For the production animals, we use the closed formula is used. It means that the details of the formulation is closed from our eyes. It means that the natural components are not exactly given. They are allowed to change in relation of the market, but should be given. This is the energy density, this is the protein percentage, the calcium, phosphate, and the salt content, and no more. The open formula is not used for production animals. It is used for pet food, for some dog and cat food to prevent allergic skin diseases. For example, the lemon rice formula is open. It's open that it contains exclusively rice and lamb meat. But for the laboratory animal science, it is good to know, not obligatory, but it's good to know the natural components exactly, because they may have secondary plant products like phytoestrogens, etc., and these compounds may influence the experiment, may have an interaction with the test material, etc. According, what about the, and here I summarize once more that yes, the laboratory animals' nutritional requirement is given according to the criteria of the reproduction and the longevity, and in the human extrapolation it is mentioned the average daily intake value, which is an important value in the human toxicology. And here is a table of the nutrients. Here I show you an example from a high-ranked article how in the experimental publication the feed and the feeding is given. All breeding mice were fed ad libitum on National Institute of Health 31 control diet from weaning, 5% weight pro weight fat, 13.5 megajoule per kilogram, A per A experimental offsprings were randomly assigned at varying to either a high-fat diet designed to mimic a Western fast food diet, equivalent to Harland diet. This is a company. 22% weight pro weight fat, which is not 5 but 40% of digestible energy, 19.4 megajoule per kilogram, or matched normal fat control, 6% weight per weight fat, 16.1 megajoule per kilogram. Feeds were obtained from and the offspring of the mice. Okay, and we will go to the picture that shows us and illustrate that the nutrients requirements can be modified by strain. Obese mice and rat, nude mouse, and here you can see the hairless guinea pig and the obese mice, or the specified pathogen-free or axenic or germ-free animals with or without microbiome. The vitamins, the degree of degradation during heat treatment. And here you can see two groups of animals with a high vitamin C requirement. Both guinea pig and fish need vitamin C supplementation. They have no gulonolactone oxidase enzyme, therefore they cannot synthesize vitamin C from glucose, and they will receive deficiency syndrome. The left side you see the prone position and shortage, atrophy of the bone development of the guinea pig, and you have see the scoliosis of the spinal cord of the fish. If you use for study the vitamin C deficiency and you use fish instead of guinea pig, this is part of the replacement. Classification of diets may be three groups. 1. Natural ingredient diet, which consists of natural feeding stuffs like in a ground form: wheat, corn, oat, fish meal, soybean meal, wheat bran, milk powder. 2. Purified diet. Many people call it semi-synthetic, but this is really purified diet. Starch, sugar, casein, vegetable oil, lard, cellulose, mineral, vitamin premix. But every time you have to add which starch, this is from potato or corn, which sugar, sugar cane or sugar from sugar beet pulp, which casein, which animal milk was used, from cow or from goat, which vegetable oil. So this is that originally they are natural products which are purified. And the number 3 is the chemically defined diet, which are the product of the chemical industry: amino acids, sugars, triglycerides, essential fatty acids, inorganic salts, and vitamins. The formulation has specific features. Should be low or no microbes in the end product. No antioxidant like ethoxymethylquinoline or butyl hydroxytoluol can be used because they can have an interaction with the test compounds. No or very low level of mycotoxins, and good availability of minerals, of vitamins. So if you have two formulations, for example zinc oxide and zinc sulfate, you have, or organic metals, you have to use the better availability form. Yes. And we have many physical forms or appearance of the diet, which is much more than in case of the production animals. One is the meal or coarse flour, which can be also used in form of cake to avoid the auto-segregation. Two is the pelleted, which has a special measure for the larger form is used for the small rodents, and the little bit smaller form is used for the rabbit. The length is half to 1 centimeter, and the diameter is 3 to 5 to 1 to 10 millimeter. The binding material is either the gelatinized starch in the cereal or added binding materials. 3. Crumbled by crushing pelleted or extruded diet and screening particle after size. 10 minutes remained. It is, for example, for one-day chicken, when the pellet would be too large to eat it. 4. Extruded by forcing wet meal, the mixture of everything, through a die under high pressure and temperature, which results in expanded product. This is used for pet when 1 liter is not more than a half kilogram. Also for fish and ape. In the next slide you will see the picture of the extruder, but here you can see that baked, all the meal and can be baked in a small laboratory use, decrease the microbial population. Here you can see the picture of the extruder, and we continue. The sixth physical form is the flaked. By mixing finely ground ingredients with water until a slurry, see when we are making a pancake, a slurry is formed, the slurry dried, and the dry sheets are broken into small particles. And these small particles will floatate on the water, in the water, and the fish and frog will think that this is a living prey. 7. Semi-moist or gel form, by adding water, agar, gelatin, or other gelling agent to meal. When toxin compounds are added, in this way the palatability is better for the animals. And the 8th is a liquid form, when we use a filter sterilization, for example, alcohol research or so. Preparation of the diets in the practice is separated in separated facility, not in the facility of the other production animals, to avoid the pre and post mixing. Rule 1. Homogeneity ground to an appropriate particle size, like 0.2, 0.4 millimeter of diameter. 2. Length of mixing time, no overmixing, no over 20-30 minutes. 3. Avoid auto-segregation during the transport, for example, and the storage and shelf time should be given. Feeding and drinking technique maybe with the exception of guinea pig ad libitum, but in chronicle toxicological research, it turned out that the ad libitum feeding gives more spontaneous tumor development and reduce the lifespan. So spontaneous tumor development and decreased expected lifespan was experienced after ad libitum feeding. The water and dry matter ratio is important, is 1 to 1.5, 1 to 2, because if the valve of the drinker does not work, not only the water consumption decreases, but also the dry matter intake. Consideration of the circadian rhythm is also important. Here you can see a picture about the ad libitum versus time-restricted feeding, and you can see that ad libitum feeding give a low amplitude of the circadian rhythm and will increase the adiposity, the glucose intolerance, the leptin resistance, the liver pathology, inflammation processes, and motor coordination. The feeding pattern, in case of time-restricted feeding, fed only during the night, and the robustness of circadian metabolic rhythm have high amplitude, but all the previously given parameters will decrease. The energetic rationing should be followed. It means that the protein level, the amino acid level, the minerals, vitamins, and the test compounds in a dry matter should relate not to the dry matter, but to the energy unit, because animals are eating according to the energy. Feeding and welfare. Welfare and environmental enrichment. We can organize that the animal should work for the feed, find from the bottom, for example, from the bedding. Lack of occupation would cause stereotypies. The solution is need to search, need to work. For that, we have to know the natural behavior. And what about the treats? In a small number of experimental animals you can use. For example, gerbil with slice of mandarin and 1 whole groundnut seed per day because of danger of obesity. Novel food items were developed for this environmental enrichment. You can see cake in original form before warming and forming or compressing, or compressed into holes in a wooden block, or cake compressed into a past shell. 4. Nutriberries. 5. Freeze-dried strawberry pieces. 6. Freeze-dried banana pieces. 7. Timothy Phleum pratense treats. And 8. Veggie treats. And you see also they really like them. Mice with cakes compressed into pasta shells and natural wooden blocks, and guinea pig with a piece of freeze-dried banana. Transport and acclimatization. You have to know the species. For example, the mini pig frequently vomit during transport, never feed before transport. In contrast, rat and mice need feed and watering during the transport. For that, the new development is the solid water, the gel-like water for transport, with or without minerals and sugar. Metabolic troubles and diseases can occur because of the not appropriate feeding. Vitamin C deficiency in guinea pig and fish. Iatrogenic dysbacteriosis can cause mostly after peroral application of large-scale antibiotics in guinea pig, but also for rabbit, mice and rats, and I also add that in case of foal, which is not a laboratory animal but important. Pregnancy toxicosis can occur, urolithiasis, susceptibility to mycotoxin. Now we have to stop for a minute. By means of the coprophagy and psychotrophy, the rodents and the rabbit once more and once more recycle part of the mycotoxins and other peroral toxins. Therefore, their LD50 value and toxicological dosage are much lower. They are prone to poisoning. And finally, in case of fiber deficiency, they use barbering and eating own hair, and zootrichobezoar or hairball can be formed in the stomach. Finally, we have to mention the brain stress microbiome, which is highly important today because we know that the brain, by means of the nervus vagus, may influence the work of the intestine, and the microbiome, by its product, the bacteria, by its secretes and interleukins and other products which are absorbed and arrive to the brain, may influence the mood and many other functions. In the human field, for example, the autism is related to the altered microbiome. And the stress between the two, because stress may change the brain function and may change the microbiome, and this is a circulus vitiosus. Thank you very much for your attention. After the lecture, I am ready for receiving your emails and your phone calls. See you in a week. It can be continued. I've seen that we have 3 minutes more, so a little bit more about the microbiome. So microbial standardization as a repetition. We have seen germ-free, mono, di, oligo, and poly-associated animals. This is not healthy at all, but useful. We have specified pathogen-free and conventional. And what is important, that there is a relation of feeding, and we have a gut-brain axis under stress, decreases the permeability of the gut wall against antigens and pathogens, slower stomach emptying, inhibition of the hippocampus proliferation by the absorbed compound, and insufficient oral tolerance can be seen. Microbiome of obese and thin mice and thin and obese human are different according a Nature publication, but the moderate stress protect against enteritis. We have seen also that the transplantation of fecal microbiota from patients with irritable bowel syndrome alters gut functions and behavior in recipient mice. And this is Irritable Bowel Syndrome article from 2017. And the Human Microbiome program is also very important. We have, before marketing bacterial product against diarrhea, eczema, ulcer, vaginosis, and for ammonia trapping in the colon. And also here stands the question, does equine metabolic syndrome affect gut microbiome? Yes. And you see that both in horse, in human and minipig, the abundance of phylum Verrucomicroba is in equine metabolic syndrome. Or there is a proposal, Lokens et al. Heterogeneity for optimizing experimental design. Here you see in Hungarian, but I will try to I try to change it. Szabványosítsuk a bélforrát. Be standardized, do standardize the microflora. And in my opinion, we need a mosaic population according to the genetic standardization's mosaic population. And the take-home message was given at the beginning till the physical form, and I have to add that we have to consider the circadian rhythm, as you have seen, but about the circadian rhythm we will have a whole lecture. Thanks very much for your precious attention.